Dear viewers, greetings. In this video, we are going to see about immunoelectrophoresis. The contents of this video contains the following topics: an introduction about immunoelectrophoresis, objectives of immunoelectrophoresis, principle of immunoelectrophoresis, materials required for immunoelectrophoresis, results of immunoelectrophoresis, applications of immunoelectrophoresis, advantages of immunoelectrophoresis, limitations of immunoelectrophoresis, and finally, precautions of immunoelectrophoresis. Immunoelectrophoresis. Immunoelectrophoresis refers to precipitation in agar under an electric field. Immunoelectrophoresis is a process of combination of immunodiffusion and electrophoresis. An antigen mixture is first separated into its component by electrophoresis and then tested by double immunodiffusion. Antigens are placed into wells cut in a gel that is without antibody and electrophorused. A trough is then cut in the gel into which antibodies are placed. The antibodies diffuse laterally to meet diffusing antigens, and lattice formation and precipitation occurs and permitting the determination of the nature of the antigens. The term immunoelectrophoresis was first coined by Graber and Williams in 1953. Objectives of immunoelectrophoresis. The first objective of the immunoelectrophoresis is separation of proteins. Immunoelectrophoresis allows the separation of proteins based on their charge and size. This helps in identifying and analyzing individual protein components within a mixture. The second objective is identification of antigens. By utilizing specific antibodies, immunoelectrophoresis enables the identification and characterization of antigens present in a protein mixture. This information is crucial for understanding the composition and properties of the sample. The third objective is quantification of antigens. Immunoelectrophoresis can be used to estimate the concentration of specific antigens in a sample by measuring the height or intensity of precipitate arcs formed during the reaction. This quantitative aspect is valuable in various research and diagnostic applications. And the fourth objective is study of antigen-antibody interactions. Immunoelectrophoresis provides insights to the interactions between antigens and antibodies. It helps in evaluating the specificity and affinity of antibodies towards a target antigen and contributing to the understanding of the immune responses and immune-related disorders. The fifth objective is characterization of immune complexes. By visualizing the lattice formation and precipitation of immune complexes, immunoelectrophoresis assists in characterizing the nature and properties of these complexes. This information is useful for studying immune reactions and diagnosing immune-related diseases. The final and sixth objective is diagnostic applications. Immunoelectrophoresis have been widely used in the chemical and clinical laboratories for the detection and diagnosis of various diseases including immunodeficiency diseases, autoimmune diseases, and certain types of cancers. It provides valuable information about the presence and characteristics of specific antigens in the patient sample. Principle of immunoelectrophoresis. When an electric current is applied to the slide layered with gel, the antigen mixture placed in wells is separated into individual antigen components according to their charge and size. Following electrophoresis, the separated antigens are reacted with specific antisera placed in a troughs which are parallel to the electrophoretic migration, and diffusion is allowed to occur. Antiserum present in the trough moves towards the antigen components and resulting in the formation of separate precipitin lines in 18 to 24 hours, each indicating the reactions between the individual proteins with its antibody. Materials required for immunoelectrophoresis. First, glass vase, conical flask, measuring cylinder and beaker are commonly used glassware item in the laboratory for preparing and handling solutions during the immunoelectrophoresis process. Reagents. Two reagents are widely used. First one is distilled water and the second one is alcohol. Distilled water. High quality distilled water is necessary for preparation of the solutions and reagents used in the electrophoresis. And second, alcohol. Usually ethanol or isopropyl alcohol is used for cleaning and sterilizing laboratory equipments and glasswares. Third material is incubator. An incubator set at a temperature of 37 degrees Celsius is essential for providing the optimal condition which is required for the incubation of antigen-antibody reaction during the immunoelectrophoresis. And the fourth material is microwave or Bunsen burner. A microwave or Bunsen burner is used for heating and melting agarose or other gel materials used for creating the gel matrix in the electrophoresis process. The fifth material is electrophoresis unit. An electrophoresis unit is required to run the electrophoretic separation of antigens in the gel matrix. It typically consists of a power supply, buffer chambers and electrode connections. Vortex mixer. A vortex mixer is used for efficient mixing and resuspension of reagents and samples. Spatula. A spatula is used for transferring solid reagents or scraping materials during the preparation of solutions or gels. Micropipette and tips. Micropipettes and disposable tips are used for accurate and precise measurement and transfer of small volumes of reagents and samples. Gel cutter. A gel cutter is used to precisely cut out specific reagents or regions of the gel after the immunoelectrophoresis procedure for further analysis or characterization. Finally, moist chamber. A moist chamber, typically a box lined with wet cotton, is used to maintain the humidity around the gel during the incubation period, preventing dehydration of the gel and ensuring optimal conditions for antigen-antibody interactions. These materials and reagents are essential for the successful execution of immunoelectrophoresis. It is important to ensure that all equipments and glasswares are clean and sterile to avoid contamination and accurate measurement and handling of reagents to obtain reliable results. Procedure of immunoelectrophoresis. The immunoelectrophoresis procedure contains 10 steps. The step 1 is preparation of agarose gel. An agarose gel is prepared on a glass slide placed in a horizontal position. The gel provides a matrix for the electrophoretic separation of antigens. The second step is well formation. Using a sample template, wells are carefully created in the application zone of the gel. These wells will be used to load the samples and controls. The third step is sample preparation. The sample is diluted by mixing 20 microliter of antigen solutions with 10 microliter of protein diluent solutions. This dilution helps to ensure the proper migration and detection of the antigens. The fourth step is sample application. Using a 5 microliter pipette, 5 microliter of the diluted sample and corresponding control are applied across each respective slit which is present on the gel. This ensures that the antigens from the samples and control are properly positioned for electrophoresis. The fifth step is electrophoresis. The gel with the loaded sample on the cathodic side is placed into the electrophoresis chamber. Electrophoresis is run for approximately 20 minutes at 100 volts. The electric current separates the antigen components based on their size and charge, causing them to migrate through the gel. The step 6 is addition of antiserum. After the completion of electrophoresis, 20 microliter of the corresponding antiserum, that is specific antibodies, is added to drops carried in a moist chamber. The gel is placed horizontally in the moist chamber and incubated for 18 to 20 hours at room temperature. During this incubation, the antiserum diffuses laterally and reacts with the separated antigens. The step 7 is drying the gel. Once the incubation period is complete, the agarose gel is carefully removed from the moist chamber and placed in a horizontal position. Blotter sheets are used to dry the gel and removing the excess moisture. The eighth step of the immunoelectrophoresis is gel washing. The gel is soaked in saline solution for 10 minutes to remove any remaining contaminants or unwanted substances. This washing steps is repeated twice or more to ensure thorough cleaning of the gel. The ninth step is drying and staining. The gel is dried at a room temperature below 70 degrees Celsius to remove residual moisture. It can then be stained with a protein staining solution for approximately 3 minutes. After staining, the gel is decolorized by immersing it in destaining solution, that is the destaining solution baths for 5 minutes. Finally, the step 10 is evaluation of results. Once the gel is dry and the staining process is complete, the results of the immunoelectrophoresis can be evaluated. The precipitin line formed indicate the specific protein antibody reactions, allowing for the identification and characterization of the antigens which are present in the original samples. Results of immunoelectrophoresis. The results of immunoelectrophoresis are obtained by analyzing the formation and characteristics of precipitin arcs or lines that indicate the interaction between antigens and antibodies. Here are some key aspects to consider where interpreting the result. It includes elliptical precipitin arcs, absence of precipitate, and identification of antigens. Elliptical precipitin arcs. The presence of elliptical precipitin arcs on the gel indicates the successful antigen-antibody interactions. These arcs represent the regions where antigens and antibodies have come together, forming a lattice structure and resulting in the precipitation of immune complexes. The elliptical shape of the arcs is typically observed due to the diffusion of antibodies and antigens during the incubation period. Absence of precipitate. If no precipitate is formed or observed, it suggests that there was no specific reaction between the antigens and antibodies. This could indicate the absence of the target antigen in the sample or insufficient concentration of antibodies to detect the antigen. Identification of antigens. Different antigens or proteins present in the sample can be identified based on various characteristics of the precipitation lines. These characteristics includes intensity, shape, and position. First, intensity. The intensity of the precipitin lines can vary and indicating different concentration or levels of the target antigens in the sample. Second, shape. The shape of the precipitin lines can provide additional information. For example, a sharp and well-defined line may indicate a strong antigen-antibody interaction, while a diffuse or smudged line could suggest weaker or multiple interactions. The third one is position. The position of the precipitin lines on the gel can help to identify specific antigens. By comparing the positions of the lines with known standards or controls, it is possible to determine the identity of the antigens which are present in the sample. Applications of immunoelectrophoresis. Immunoelectrophoresis has a wide range of applications in various fields, particularly in medical diagnostics and immunology. Here are some key applications of immunoelectrophoresis. It includes identification and quantification of proteins, diagnosis of gammopathies, monitoring antigen and antibody purity, qualitative analysis of M proteins, and finally, evaluation of therapeutic response and immune system disorders. Advantages of immunoelectrophoresis. Immunoelectrophoresis offers several advantages as a powerful analytical technique for the identification and characterization of antigens. Immunoelectrophoresis offers several advantages including high resolving power, the ability to identify multiple antigens in a sample, sensitivity, versatility, and cost effectiveness. These advantages have made it a valuable tool in various fields, including medical diagnostics, immunology research, and protein analysis, and also contributing to our understanding of antigens and their interaction with antibodies. Limitations of immunoelectrophoresis. The first limitation is slower and less sensitive when compared to immunofixation electrophoresis. The second limitation is limited detection of small monoclonal M proteins. And the third limitation is limited availability of specific antibodies in food analysis. Precautions of immunoelectrophoresis. Precautions need to be taken during the immunoelectrophoresis experiment to ensure accurate and reliable results. Here are some key precautions to consider. First one is familiarize yourself with the procedure. Second, wear gloves. Third, proper preparation of reagents. Fourth, clean glass plates. Fifth, neatly cut wells and troughs. Sixth, temperature control. Allow the agarose gel to cool to 55 degrees Celsius before adding to the antiserum because high temperatures can lead to the inactivation of antibodies. Finally, maintain a humid atmosphere to ensure that the moist chamber contains enough wet cotton to maintain a humid environment. This helps to prevent the drying of the agarose gel during the incubation and ensures optimal diffusion of antibodies and antigens. By following these precautions, you can minimize the risk of contamination and maintain the integrity of reagents and create suitable conditions for accurate immunoelectrophoresis results. Dear viewers, that's all about the immunoelectrophoresis. Thank you for your support. Thank you.